So there's two kinds of fractionation. One is where everything's sort of random, and one where it's all on one, but not the other. As if when the two genomes came together, one genome was a way better target than the other. And that turns out to be the case in Arabidopsis. My lab found that a while ago. And it turns out to be the case in maize as well, and maizegenome.org. And my lab found it. It's so obvious, as you'll see. I hope you can see this, because this is a simple dot plot out of one of the just comparing the maize genome to the sorghum genome, and you just put it into our little SynMap algorithm, and you press color code for KS. So what we're doing is for any gene pair, it's changed into proteins aligned, kind of, and you do a KS or a third position frequency of base substitution measure and color it. And so you can see whether it's kind of recent, that would be purple, or kind of a line that was, that's on your thing. Awfully colored. Kind of a line like that, which is a blue line rather than a purple line, and it's the purple lines that are the maize sorghum syntenic lines. These are gene pairs that are collinear. And if this is leftover, these little blue lines are leftover from the pre-grass tetraploidy. If you don't have this color code, it's sort of hard to tell. And of course, the maize has had a duplication and a fractionation, so let's look at this. This is sorghum. So for one sorghum chromosome, let's say chromosome 1, there should be two maize chromosomes. Well, there's one of them, maize 1, and the other one is there, and oh, there's the other arm, there. And let's say maize chromosome there, and maybe there, and there, and there is one, and that fractured thing is the other one. You can see a lot on this, especially when you get rid of the syntenic lines that are from an older or previous event. It all makes sense. It's so clearly a tetraploid. And it's easy to see, for example, that if you compare this region of this is maize, so this would be the short arm of, no, this would be the long arm of chromosome 2 with the long arm of chromosome 7, that those would be homeologous. Those would be the two paired regions, that and that would go along with the sorghum region. I found 37 of those regions in vivid detail using the tools in Koji. If you want to find our toolbox, go to Koji. Just go to Google and go to Koji. Where we mask everything in sorghum except what it shares with rice and what's called an exon. And then we only look at what happens to those shared genes in maize after the duplication. And we compare it to the panels, which are repeat masks, that's purple. The orange is turned to N or masked, everything but the genes. And we get a stretch of models. We only look at those models that are shared. We throw out the duplicates. We throw out the insertions into maize, which there are many. We're just asking for what happens to the shared genes. And the shared genes look something like this when we're done with these 37 regions. They're both there. One's there, the other one's there, one's there, one's there, both, one, both, the other, both, one, the other, both. And we have 37 regions like this, and we cover, roughly speaking, 20% of the genome. We'll cover the rest in another way, by angles of syntenic lines. But we cover these by manually looking at every gene. And then when something is present as both, we evaluate whether that means it's actually completely there, and we find that a good 10% of the time, it's not completely there, there's a bite in it. So it goes along, and then one of the exons is sitting there, and there's a big bite taken out. Fractionation is recent in maize. It's not very long ago. I think it still has the bite marks of the mechanism that's removing things. That was the idea. So when we look at all of these regions, what we find, roughly speaking, is that 68% of them are exceedingly biased. 32% are not significantly biased, which means that it looks like a whole genome is attacked by whatever mechanism removes genes, 2.3 times more than the other. The mechanism, by and large, here's runs of genes and the frequency of that run. Here's a run of one gene lost, 2 genes lost in a row, 3 genes, 4 genes. This is basic coin flip kind of stuff. And this is the 95% confidence intervals around the simulation that 1 gene at a time is removed. And basically it's close, but no cigar. We couldn't help it much if we got rid of the biggest and strangest long deletions. There are some very long deletions. Some of them are actually segmental transpositions. We get closer, but we can't. The only way we can get it to be perfect is to make it 80% 1 gene, 15% 2 genes, and 5% 3 gene deletion mechanism. And then everything's perfect. Runs are similar. If we take runs, we still get essentially the expected curve based on 1 gene lost at a time. But if we correct for the 80%, we used an evolutionary algorithm to simply find out the distribution that would make the experiment fit. Just make it fit. And this is it. This is the evolved answer. Doesn't mean it's right or anything. It's circular. But when we use the same 80-15-5, then the runs of retentions also fit expectations. Roughly speaking, it looks like it's 1 gene at a time. That doesn't mean it's really 1 whole gene. It means it's not 2 genes or 3 genes. The deletion mechanism is tiny. We know it's deletion because when we look, we see deletions themselves, little bites, and around them tend to be flanking direct repeats. This has been seen before in transposons by Devos in the Bennetson lab, looking at transposons, and Petrov, right here from Stanford, looking at Drosophila transposons, and we now find it true for genes, not just transposons. The mechanism looks something like this. The chromosome can make a circle like this. A fellow called John Wannon, a long time ago, worked on bari, a duplication in Drosophila, and other large tandem repeats and their instability, and found circles like this, and you get a normal recombination and off comes the circle. That's a perfect example where you just always be searching to make little loops based on just randomly occurring flanking repeats and get a recombination there. That would explain the kind of sequences we see around the bites in the DNA. And I see the nucleus as being a cauldron of circles. Just circles everywhere. Sometimes they might go back into the chromosome and it gets a fragment, but mostly they just go away and eaten. Cauldrons. Animals don't do this. They kill genes in situ and they wander around as pseudogenes, open reedy frames with breaks in them. That's not what plants do. Plants remove their genes, and the only pseudogenes you see in the animal sense are in the centromeric regions. Presumably they can't do this. They can't get eaten. Anyway, that would be my guess. And why plants? I'm sorry. Go, do it. Why plants? Because plants can afford to carry genetic load. They get a lot of pollen grains. And what's wrong with losing a few percent of them? They have a lot of seedlings. So you lose 10% of them. It's not like a chimp. You don't have many babies. You don't have any gametophytes. You can't carry load like a plant can. The population geneticists generally hate me to talk like this. But there's no reason you have to have a formula to talk about populations. There's no rule.